All right, folks, it's time to get going here. The slide behind me says it all, right? The next time you guys show up in this room, or if you're one of the students taking the online section, excuse me, in 101 Fawcett, what we'll be doing is giving you a test, right? So this is the same slide I've had up the last two lectures as well. Nothing too surprising, I hope. 45 multiple choice questions, just on things that I've talked with you about in class, and bring a pencil. That's pretty much all that I think you need to know at this point in time. The test started in its infancy as the one that I gave last year to the fall 2015 version of the Bio 1120 class. I copied it, and then I went to work on changing it around a little bit. So if I were you, I'd start by looking at that old test, and that'll give you a good feel for the way that I'm phrasing questions and the breadth of the questions that I will be asking you on Thursday. Just as something of an aside for you, I'm not going to be here on Thursday myself, but we'll have the supplemental instructors and the graduate teaching assistants from the laboratory around to hand out the tests for you. And the plan is for them to be graded and the grades posted on Pilot that same day. So sometime on Thursday afternoon, that should all be taken care of for you. Excuse me. I'm going to be giving some talks in California, and if you have questions or really need to get a hold of me, send me an email, and I should be able to get back to you relatively quickly. There's a 3-hour difference, and so when you guys finish the test is before people over there start waking up, and I should have no trouble getting back to you right away. All right? That means that I won't be around for my office hours on Thursday morning, but it also means if you send me an email with some questions, I'll be able to respond to you pretty quickly, and I think the supplemental instructors will be available to field questions from you outside my office during my office hours. And then also there's an online review session tonight. Look to the Pilot page of the course. Was the time 8 till 9? It's on Wednesday, 8 till 9. All right, not tonight, Wednesday night, online, using Pilot Live. And you'll be able to get to that through the Tegrity website. So if you have questions, that's sort of a review session that you could use, and I think you might find that helpful as well. Any questions for me about the test? You guys are veterans now. It should be no big deal to you. All right, so let's start talking about some fun stuff. You know what, for today, for the very first time, I finally get to talk with you about genetics. The name of this course is Cells, Genes, and Genetics. Thank goodness we're moving into the genetics part of the course. It's the genetics stuff that got me all fired up to become a biologist in the first place, right? And so I've been looking forward to having a chance to talk with you about these things since before this class has started. This is good stuff. You'll see that at the top of the outline, really the new stuff that I'm going to talk with you about today is Mendelian genetics. And that's a tip of the hat to an Austrian monk whose name was Gregor Mendel. Gregor Mendel figured out most of what I'm going to be talking with you about today, and I'll tell you the couple things that he wasn't able to figure out along the way. But he figured out these things in the mid to late 1800s. So a lot of what it is that we'll be talking about today, the cornerstone of genetics, are things that humankind has really only wrapped their heads around for about 100, maybe 150 years. So this is still relatively current stuff, relatively new stuff. We take Mendelian genetics for granted. It just seems so obvious. You know, I've got a 6th grader at home, and she's explaining dominance and recessiveness to me. I don't know what they're teaching them in the Bellbrook High School, but or the middle school, but wow, you know, it's amazing to me that it's permeated through, excuse me, so many different grade levels that this stuff gets taken for granted. But I want to take you by the hand a little bit today, make sure we're all on the same page and all using the same words, And then with that foundation, after you're done with the test on Thursday, we'll start talking about some of the higher level implications and applications of these insights. But again, it starts with this Austrian monk, Gregor Mendel. And I gotta tell you, I'm a fan of Mendel, all right? He's an admirable individual. Something that he may not get as much credit for as I think that he should is he was an exceptionally humble individual. He had a lot of humility. It's maybe not too surprising. For goodness sakes, he was a monk, right? You kind of expect monks to be salt of the earth, good, upstanding citizens, but he was really very humble. And his humility helped him enormously in cracking the nut that was genetics and that we now understand to be what Mendelian genetics is all about. Again, it's his humility that played a really important role in allowing him to make the advances and get the insights that he ended up getting. For thousands of years, people had been trying to figure out how it is that information got passed from one generation to the next. It's been something that mankind has been curious about, keenly interested in for thousands of years. Probably close, the thinking, the curiosity probably began close to the time that the discovery was made that sex gave rise to progeny. Right? Can you imagine, at some point in our distant past, Bronze Age or something like that, people would have had to have figured that out. Like, oh, hey, what happened over there? And then after that light bulb went on, why do my kids all have big noses? What's the deal there? What's happening with all that? Gregor Mendel recognized that. He was a student of Aristotle and Plato. That was part of his training. And he recognized that prodigious, legendary thinkers, deep philosophers like Aristotle and Plato had tried to figure out how it is that like begets like. That was the phrase that they would often use to describe it. Why is it that your children look so much like you? What's the deal with that? And they had some crazy ideas. They had ideas like inside of every sperm was a miniature version of the guy that when it would bump into an egg, would bump into a miniature version of the woman, and the two would kind of blend together into some new thing, right? I mean, they had some really crazy ideas. But in the end, they really didn't have much of a clue aside from being able to say, like begets like. They couldn't answer that question as to how. And again, Mendel was a student of Aristotle and Plato and these other great thinkers. And he came into this whole thing saying, gee, I'd like to know why it is like begets like, but I'm just a humble monk. How could I possibly hope to figure something out that Aristotle and Plato and others have wrestled with and never been able to really get a satisfying answer to? You know what? Mendel's thinking to himself, I'm just going to I'm going to set the bar lower. I'm going to set my goals a lot lower than what they did. They're trying to figure out something noble and lofty like human inheritance. Mendel comes along and he says, I'm just going to work with something very simple. I'm going to work with pea plants. All right, so he chooses pea plants as his model organism to figure out inheritance in. He doesn't even begin to think that he's going to figure out what's going on with humans, let alone something like cats and dogs and horses and sheep. He's just setting his sights on pea plants. Maybe he can learn something just to satisfy his curiosity about how it is that inheritance happens in something simple like a pea plant. And in that humility, setting the bar, the goal kind of low, was actual genius. He may not have actually been a genius, but boy, that was a stroke of enormous luck for him to be able to figure this stuff out by focusing on an organism like pea plants. Because pea plants are a heck of a lot better model genetic organism than things like cats and dogs and chickens and humans are. Let me give you some of the things that make pea plants a really good model organism. There's a number of things. One of them is they have lots of progeny, right? So he can see, he gets large numbers that he can then do some simple statistical analyses on, right? With his pea plants, you'll see in a few slides that his very first mating experiments with pea plants gave rise to on the order of 1,000 baby pea plants, right? If he was trying to do the same sort of thing with humans, that's just an absolutely unrealistic expectation that you're going to have thousands of children from a particular mating experiment, right? And you're forced, when you're doing human genetics, to base all your conclusions on much, much smaller numbers of offspring, right? And that really compromises how confident you can be in the conclusions that you draw. With pea plants, you can get thousands of progeny. Another big advantage to pea plants over the other organisms people had been trying to figure out before Mendel is not only do you get lots of progeny, but you get them really quickly. Mendel was able to get 4 or 5 generations of pea plants every year, okay? Compared to a human mating experiment, for instance, where my daughters aren't going to be even dating until they're in their mid-20s. So what are the chances of having children? It's going to be forever before, it's going to be decades before my kids start having kids, right? I need some assurance on that. But it's going to be decades before my kids start having kids. But with pea plants, you can get generations every 2 or 3 or 4 months, right? You can have short generation times, so you can do many more experiments in the same amount of time that you would have been able to do if you had used a less good model genetic organism, like humans, that have small number of progeny and take a long time for those generations to cycle to the next generation. Let me also draw attention, as a father, to the cost of upkeep, right? You know how much it cost Mendel to keep his pea plants growing? It was literally pennies. He'd go down to the farmer supply store in his village, and he would pay a few pennies to buy a bag full of seeds, and off he'd go, and he'd be able to do all of his work. That's very different for even a dog in terms of mating experiments. Small number of progeny, long generation times, and they need to eat, right? And they need to be housed, and they need veterinary care, for the dogs at least. For children, oh my gosh, it's 10 times, 100 times those types of costs. And so Mendel, with his pea plants, has large numbers of progeny, short generation time, small cost of upkeep. And then the last one turns out to also be a very helpful trick. He was able to tap into an existing set of plants that had already been characterized for a number of different characters, different features, things like the flower color. He could go to his supply store and he could buy pea plants that only flowered purple when they developed flowers. Or he could buy pea plants that only flowered white when they developed flowers. He could buy seeds that would give rise to pea plants that were tall. He could buy seeds that gave rise to short pea plants. He could buy seeds that gave rise to yellow peas, or to green peas, or to smooth peas, or to wrinkled peas, right? He had a number of different characters, 6 or so, that he could just tap right into and get off to work with. Whereas with humans, or other model systems that others had tried to figure out in the past, the water was murkier. Hair color, gee, it's not really just a matter of blonde, brunette, and redhead, right? There's gradations in between. What's the character that you're looking for there? Height, oh my gosh, there's such a range of heights. It's not like you have clear demarcations. The waters were much muddier in these more complicated organisms that others had tried to study before Mendel and came up with nothing beyond like begets like. But again, Mendel, in his humility, says, I'm just going to see if I can't figure stuff out with these simple little plants, these pea plants. They're cute. I like peas. I eat them. It's good, right? Let's see if I can't figure some things out on the side. I'm growing the peas anyway in my garden. May as well see if I can't learn something along the way. And for whatever reason, genius, luck, whatever, he stumbles upon a really good model genetic organism. Excuse me. And so, what does Mendel do? He does some mating experiments. Okay? You can't do genetics. And as a geneticist, I'm saying this to you very authoritatively, you cannot do genetics unless you do mating experiments. That's at the core, it's at the heart of everything involving genetics. You're looking strictly at how it is that information gets passed from one generation to another. With sexually reproducing organisms, part and parcel with that passage from one generation to another, to the next, is sexual reproduction. So you need to have mating experiments, and sure enough, right out of the gate, Mendel starts to do some mating experiments. The picture on the screen right now is, in fact, a cartoon of sorts that tells you what it is that Mendel's very first mating experiment was. He went down to his farmer's supply shop. He picked up some seeds. The guy behind the counter told him, hey, check it out. I got this bucket of seeds here that's going to give you nothing but purple flowering plants. And I got this bucket of seeds that's going to give you nothing but white flowering plants. You want to decorate your abbey a little bit with purple? Well, these are the ones for you. You want to decorate it with white? These are the ones for you. Take your pick, right? And Mendel took a little bit of both. He took some true breeding purple flowering plants and some true breeding white flowering plants. There's a technical term I slipped in there. It's the same term that was used at the time. True breeding, simple, right? It just means that if you let those purple flowering plants breed amongst themselves, you'll never get anything but purple flowering plants at the end of the process. And if you took the white flowering true breeding plants, same thing. If you let them breed amongst themselves, you get nothing but white flowering plants. So he's got true breeding purple and white flowering plants. And here's a little more jargon for you. Again, I want to make sure we're all on the same page with the terms that are being used at this point. True breeding, got it. Character. What he's decided to do with his first experiment is focus on a single character. Okay, the character is flower color, and he's got two different traits that he's keeping his eye on. The purple flowering trait and the white flowering trait. Okay? So a character is just something for which you can detect differences, and then the trait are the manifestations of those particular differences. Okay? Flower color here is the character. Purple is one trait. White is the other trait. And again, Mendel sets off to do a mating experiment. And he wants to know, what do you suppose would happen if I breed a purple flowering plant with a white flowering plant? Again, stop and think for a moment. What a simple, humble kind of question. Nothing lofty like, what happens if I make that smart woman have children with that dumb guy? Boy, that's going to be messy in a whole bunch of different ways. He's asking something very simple, not a big deal. Nobody's going to write a newspaper story about this. What happens if I mate a purple flowering plant with a white flowering plant? What kind of little baby pea plants are they going to have? Curious guys, curious monks wanted to know. And so that's what he does. He cuts off the pollen-producing cells from a purple flowering plant. He leaves the egg cells behind, and he uses a paintbrush to transfer pollen from a white flowering plant to a purple flowering plant. It would have been fine if he had switched the sexes around here, would have gotten the same result. But at the end of this mating experiment that he set up, he gets some peas. And that then is going to become what he grows up then to be the second generation that will answer his question of what's going to happen when I mate a white plant with a purple plant. What kind of baby pea plants am I going to see? One last bit of jargon on this slide. Notice this is the pea generation. So this plant and this plant are what are referred to as the pea generation, not because we're talking about pea plants, but as you can tell from the slide, because we're talking about the parental generation. And it's unusual for people to bother to spell out parental to you. Your textbook, and I suppose from here on out, are simply going to be talking about the P generation. This is the generation where we've taken true breeding plants or true breeding individuals. We've had them mate with each other, and we want to see what kind of babies, what kind of progeny they have. That's the P generation. In the same vein, these babies here are the first set of progeny that come out of them, and those get called the first filial generation, or the F1 generation. You guys know who named the city of Philadelphia? It was Ben Franklin's idea to come up with the name Philadelphia. And it was Ben Franklin who thought Philadelphia would be a neat name for a city because he knew that filial or philia meant brotherly, and what he wanted is that Philadelphia to be the city of brotherly love. That was his explicit thinking behind coming up with the name Philadelphia for the city that still exists today on the coast of Pennsylvania. And so in the same sense that Franklin was talking about Philadelphia, filial, brotherly, that's where filial comes from. It's the first generation of siblings that come out of this family. And again, it's the first generation, so it's the first filial. People rarely bother to talk about first filial. More typically, you'll hear them refer to the F1 generation. Again, short for first filial. And this is a cartoon version of the results that Mendel got from that particular mating experiment. I remind you, he started with purple flowering plants and he started with white flowering plants. He mated them and he wants to know what kind of baby pea plants will he see. Waits a couple of months, they grow up, and what does he find? Every darn last pea plant that came as a result of that mating experiment, every one of them was purple flowering. It's the sort of thing that would make a monk sort of scratch his head and say, what's up with that? You know, I'm sure I had some white flowering plants as half of the parents. What happened? Why is it only purple? And he begins to start thinking, as I think any of us would, there's something about whatever's determining the purple flower color that wins in a fight against the white flowering thing. Whatever that thing is, purple seems to be dominant, that's the word, in some way to the white trait, which is maybe recessive. And at this point in time, I think a lot of us probably would have stopped. It was a trivial question. He got a trivial answer, a little bit of a puzzling answer perhaps. Excuse me. But he took it one step further. He ended up having these pea plants from the first filial generation mate amongst themselves, and then asked, and again, this is another issue that makes pea plants a good model genetic organism. There aren't any ethical constraints that come from these mating experiments. Can you imagine a human geneticist doing the same thing? Oh, it's a nice family you got there. Let's have them have sex amongst themselves so that we can see what kind of progeny they have. Again, pea plants, brilliant, or at least an extremely lucky choice on Mendel's part for a model genetic organism. And again, Mendel says, what's going to happen if these guys are left to breed or mate amongst themselves? What kind of progeny will you see? And he's, got to be speculating. What's going to happen? When I mated a purple flowering plant with a white flowering plant, I got nothing but purple. Now I'm mating purple flowering plants with other purple flowering plants. Maybe what's going to happen is I'm going to see nothing but purple all the way down. As many times as I do this mating experiment, it's going to be purple, right? That's one possible explanation. Could be that. Maybe, if I keep doing this long enough, sometime I'm going to find white flowering plants again. Could happen. White flowering plants have to come from somewhere in the first place. Maybe if I do this enough times, I'll start to see white flowering plants again. And so he does this mating experiment, an F1 cross, F1 mating experiment where he had these plants mate amongst themselves. And again, if sixth graders know about this, I suspect this isn't going to be a big reveal to you, but when he does that P generation mating experiment, he gets only purple flowering plants, and when the F1 cross takes place, he gets an F2 generation, the second filial generation, where, dare I say it, lo and behold, the white flowering pea plants emerge again. He starts to see some white flowering plants one more time. So the white flowering thing, whatever caused the white flowering plants in the parental generation to be white flowering, has been faithfully transmitted across 2 generations. You couldn't see any indication of it in that first generation, but in the second generation, there it is again. But there's this little bit of an interesting twist as well. Because he has literally on the order of 1,000 different F2 plants to look at, he can be really confident in saying he is not getting equal numbers of purple flowering and white flowering plants in that F2 generation. Instead, there's an interesting ratio. It's about a 3-to-1 ratio of purple flowering plants to white flowering plants. And these are the actual numbers that Mendel recorded in his diary, his lab notebook of sorts, as he was doing these experiments. How about that? Couple of things that are kind of worth maybe just noticing to make this perhaps a little bit more interesting. And Mendel wrote those numbers down. This is the very first mating experiment that he did, but it must have taken him, what, a half hour to count all of these different baby pea plants? Oh, is this one purple or white? This one purple? It took him some time, right? And he was interested in the result, but he was also interested in the specifics of the result. He must have looked at his garden and said, wow, that's a lot of purple flowering plants. Hey, look at that, there's some white ones back again. That's cool. There seems to be a lot more purple than white. How many more purple than white? He took the time to actually count them, and he took the time to write them down right out of the gate. So despite his humility, and despite his own thinking that this is a fairly trivial, unimportant question, he's taken it pretty darn seriously right out of the gate, not particularly well trained in science. He's a monk. That wasn't his job to be a scientist of any sort. And he writes these numbers down, and he notices it's about a 3-to-1 ratio of the purple trait relative to the white trait. That 3-to-1 ratio has come to be known as a classic Mendelian ratio. 1-to-1, 1-to-0, 3-to-1, and later on you'll see 9-to-3 to 3-to-1. These are ratios that say something that now, when a geneticist sees a 3-to-1 ratio, is like, I know what's going on, right off the top, right? Mendel still had to work these things out. We'll talk about that in the next couple of slides. But that 3-to-1 ratio is a classic Mendelian ratio. It's fairly typically encountered when you're doing mating experiments. So, vocabulary sorts of things. Just once again, the first generation had a character that he was focused on, flower color. There were 2 traits that he had available to him to do experiments with, the purple trait and the white trait. He took some of those plants, had a P generation, a parental generation. It's important to remember that when he bought them from the store, these were true breeding, meaning the purple flowering plants would always give rise to purple flowering plants, and the white flowering plants would always give rise to white flowering plants. But then he does a mating experiment between them. He gets a first filial generation, an F1. He finds that all of them display the dominant trait, the purple flower color for this character. And then when he does this curious thing of doing a F1 cross, he mates one F1 plant against other F1 plants. He gets a second filial generation, an F2 generation. And in the F2 generation, he gets a 3-to-1 ratio of purple flowering, dominant flower color versus white flowering, recessive flower color. Okay? So that's it all in a nutshell. Well, this was just the start for Mendel. It literally was the start. He started with the purple and the white flowering plants, and then if you look at his diary, his notebook, you'll see he didn't stop there. It got him fired up. He went back to the farmer supply store in his village and he says, hey, what other kind of pea plants do you have? Do you have any other characters that I might be able to distinguish between one pea plant and the next? And they were very obliging and they said, hey, do you want to have flowers that grow in the middle of the stalks for the pea plants, or do you want flowers at the ends of the stalks? Because I can get you seeds that do one or the other. And do you want to have yellow peas, or do you want to have green peas? I can get you either. Do you want to have round peas, or do you want to have wrinkled peas? Either. Do you want to have puffed up pea pods, or do you want to have shriveled up constricted pea pods? Because I can get you seeds that give rise to either of those. Do you want to have green pea pods or yellow pea pods? It doesn't matter what color the seeds are. I can get you different colors of pods. And do you want to have tall pea plants, or do you want to have little shrimpy pea plants? What kind do you like? Mendel says, I'll take them all. Right? Here's my quarter, right? I'm going to buy a little bit of all of your different kinds of pea plants. And now he's got, let's count them together, 1, 2, 3, 4, 5, 6, 7 different characters that he can study. He starts with the flower color character, and now he's added 6 more to his list. And he does monohybrid crosses with all of them. Okay? Hybrid, meaning he's doing some mating experiments. Monohybrid, meaning that he's just keeping his eye on one character, like flower color or seed color. He's not doing 2 at the same time or 3 at the same time, right? It may be that these are all short pea plants that he's tracking here, but he's just keeping his eye on something like the seed color. It's a monohybrid cross, and he does 7 different kinds of monohybrid crosses. He had already gotten data, numbers that he bothered to record for the flower color character. He sees that's a 3.15 to 1 ratio. Come on, between friends, that's a 3 to 1 ratio. All right? Then he gets data for these other characters. For the flower position, he finds that middle of the plant seems to be the equivalent of purple in the sense that it's dominant. You get a 3 to 1 ratio of middle flowering plants to end of the branch flowering plants. If I were to ask you, and you forgot what it says up here, what do you think? Is this dominant or recessive? Well, a real good clue would come if I told you that when he mated true breeding axial flowering plants with true breeding terminal flowering plants, all of the progeny were axial flowering plants. All right? That's a really good clue that axial flowering is dominant and that terminal flowering is recessive. All right? And then the next clue comes from the fact that if you did mating experiments with those F1 pea plants, you would get a 3 to 1 ratio of axial flowering to terminal flowering pea plants at the end of that process. Again, 651 axial flowering to 207 terminal. And again, he's not stopping there. He's keeping his eye on seed color. He's keeping his eye on seed shape and these other traits as well. And he records all of these numbers. Look at this one here. This was one that apparently he was really interested in. He wanted to beat this one up hard. He got 6,000 yellow-seeded pea plants in his F2 generation, and he got 2,000 green-seeded pea plants in his F2 generation. Here's another one that he must have spent a couple growing seasons on getting these numbers, seed shape, round versus wrinkled. But time and again, he's getting these numbers that are consistently telling him 3 to 1. The dominant trait in the F2 generation keeps showing up in a 3 to 1 ratio. And at the end of this, he's getting the message loud and clear. Something is going on. And he starts to formulate some hypotheses. He's thinking, what could possibly explain this? And the answer he comes up with turns out to be what we know now to be the right answer. It's the only thing that made sense to him. The answer that he comes up with is that these pea plants, every last pea plant, must have two sets of instructions for each of these characters that I'm studying. They must have two sets of instructions. We've already talked about it in this class. These are diploid organisms. Mendel didn't know about diploid or tetraploid or haploid. That stuff hadn't been worked out at that point in time. But he comes up with this idea, they must have two sets of instructions. And when the purple instructions for flower color are in the same context, the same organism as the white flowering instructions, the purple must win. That explains what he sees. But the white one is still there. The white instructions are there. It's just you can't see it because the purple one is such a dominant trait compared to the white. So there are two instructions. They're both in the cells, or he didn't think about cells so much as in the organism. They're both in the organism at the same time. One of them manifests itself in the appearance of the organism. The other one doesn't, but it's still there. And when the two are together in a single organism, they don't blend or hybridize or smush into each other in any way because those instructions can get faithfully passed on in a subsequent generation without having changed, even though one was in the presence of the other set of instructions. What he comes up with is this idea of particulate inheritance, that the character flower color is governed by a pair of particles. One of those particles gives you the purple trait, the other gives you the white trait, but those particles, two, have to be in each of the organisms, and when they're particulates, they're not blending or bumping into each other in any way, they're just hanging out together, and later on in another generation, they can go their separate ways. Particulate inheritance. And that idea of two particles and particulate inheritance was it. There was nothing else to do after that, because with that in mind, all of his results made a lot of sense, and he seriously could not think of any alternative that explained the kinds of numbers that he was getting with these pea plants. So that's what this is all about, particulate inheritance. We've talked about characters, we've talked about traits, flower color, seed color, those are characters, purple versus white, green versus yellow, those are traits. Hybridization is just fancy genetics talk for mating, for getting these two different kinds of particles to come in contact with each other. And then there's this sort of subtext that there are two particles in each organism and that the particles remain somehow separate. They don't blend together in any way as they're being passed from one generation to another. And all of that then delivers to Mendel this insight that now we call the law of segregation. It's essentially the law of segregation. We're just talking about the fact that these particles get separated from each other when gametes are being made. When are gametes made? Gametes are made during meiosis. That's what we talked about all class on Tuesday. During meiosis, these particles separate from each other. Now, let me skip ahead in some sense here, because remember, Mendel's doing this stuff in the mid to late 1800s. DNA wasn't recognized as being the genetic material until 1953, okay? And chromosomes were just vaguely understood. People who really studied with microscopes a lot could tell you, oh yeah, there are these things, these structures called chromosomes inside of cells. And I'll tell you, the people who talked about chromosomes back then probably didn't attach any more significance or have any greater insight in terms of what a chromosome was or what was important about a chromosome than you guys might have about, oh, hey, that's a mitochondria, right? You know, this is a big deal. It's just a feature. It's one of the things you notice when you're looking hard inside of a cell. Not a big deal. So Mendel doesn't know about DNA. He doesn't know about chromosomes. But he comes up with this idea of particles. And now we can relate those particles to what we do appreciate about DNA and chromosomes. What's actually going on with the flower color in today's jargon is that there are two different alleles for the flower coloring gene. The reason there are two traits is because there are two alleles. An allele is a chunk of DNA containing some information that's recognizably different than an equivalent but nonetheless different chunk. So along the length of one of the pea plant chromosomes is a stretch of information that determines flower color. If you have the purple flowering version of this gene, if you have that allele, you have one particular set of instructions at that spot on the chromosome. If you have the white flowering allele, you have a very similar but subtly different version, and that subtle difference in turn gives rise to white flowering plants as opposed to purple flowering plants. So again, an allele is a recognizably different variant at the DNA level between one version of a gene and another version of a gene. Those genes are nucleotide sequences that are embedded within the length of a chromosome, and the particles that Mendel was talking about and thinking about, the particles, you could think of them either as the gene. This could be one particle here or one particle here. Or if you prefer, it's no great sin to think of the particle as being the chromosomes, these two copies of this particular chromosome. One of these copies would have come from the individual's mother. The other would have come from their father. We can't tell which is which from what we're seeing here, but each parent would contribute one chromosome. One of those might have the purple flowering allele. The other may have the white flowering allele. Because again, we're diploid organisms. We have two copies of all of our genetic instructions. We get half of those instructions from mom and half of those instructions from dad. Diploid, that's what it's all about. So Mendel didn't use any of these words. He didn't know about chromosomes. If he did, he didn't think they were particularly important. He had no concept of what a homologous chromosome was, like you guys do now. He didn't really appreciate that DNA was playing an important role in all of this, and he never bothered to use the word allele. He just kept focusing on characters, traits, and particles. So let's see how this law of segregation plays out. Okay? There's a little more jargon for you on this slide as well. So what we're seeing here are the results of that first F2 generation that Mendel had to work with. Okay? Flower colors, the character, purple, white are the two traits. His first F2 that he's looking at, the numbers are scrubbed away here, but you can see there's that 3-to-1 ratio of purple flowering ones to white flowering ones on the left. So let's talk about a couple of new words, perhaps, on this slide. This idea of phenotype. Phenotype, very simply, is what the organism looks like. Okay? Say no more. That's what phenotype is. It's what the organism actually looks like, what trait is actually being displayed. Genotype is a much more modern concept. Mendel didn't really have the capacity to talk about genotypes, but genotype is a description of what kind of alleles the individual organism has. Right? So the phenotype is what trait is being displayed. The genotype are the combination of alleles that are associated with the organism that you're talking about. There are two different kinds of genotypes you might get at a single gene. You could either find that the genotype is for a homozygote or for a heterozygote. Let's start with hetero. You guys know what heterosexuals are, right? All right, these are individuals that like to have sex with people of the opposite sex. Hetero meaning two different things are having sex, okay? Heterozygotes are the organisms where for the locus, the gene that you're talking about, there are two different alleles. So a heterozygote for the flower color gene would be an individual, an organism that had one copy of the dominant purple gene and one copy of the recessive white gene. And that's in contrast then to homozygotes. Homosexuals are individuals that like to have sex with people of the same sex that they are. Homo, same, sex, there you go. Put them together, you got homosexual. Homozygote means that within this organism, the genes for that particular, or the alleles for this particular gene are the same. Here, they're abbreviated capital P and capital P. As a fairly good shorthand and nomenclature, the name of a gene is usually the first letter of the dominant trait. Okay, so here capital P comes from the fact that purple was dominant to white. Okay, so two uppercase P's means that what we see here is an individual that has two copies of the purple allele. But there's another kind of homozygote that you might get. These are the ones that have two copies of the recessive allele. Again, by convention, typically what you'll find is the dominant trait is what gets the letter in the first place. Uppercase means dominant, lowercase means recessive. Two lowercase p's is a homozygote for the white flowering allele. Now, what kind of pea plant is this going to be? You know the answer, right? It's got nothing but white flowering alleles in it. It can't do anything but make white flowers on these pea plants. What kind of pea plant is this going to be? If you have that genotype, you got two copies of the purple flowering allele. Come on, there's no choice. You can't do anything but make purple flowers. So the homozygotes, it's pretty easy to make sense of what's going to happen there. What's going to happen with these guys? The heterozygotes. One gene for purple flower, one gene for white flower. We've gone through this a couple times already at this point in the lecture. Purple is dominant to white. If there's a purple gene in the organism, you're going to make purple flowers. If you have two copies of the purple gene, you're going to make purple flowers. If you got one copy, you're going to make purple flowers. If you got no copy, you're making white flowers. It's pretty much what it comes down to. So, Mendel can see 3 purple flowering plants for every white flowering plant in his garden. We know the genotype of the white flowering plants, right? There's no mystery about that. They're an open book. We know exactly what their genotype is. If there was a purple allele in these plants, they wouldn't be white, right? They are white, therefore they must be homozygous for the recessive allele. But if you pulled a random purple flowering plant from the garden, you'd kind of have to just wonder, is this one of those purple flowering plants that's the homozygote for the purple gene, or is it one of the heterozygotes? And the best that Mendel could do, if you set it up to him that way, is say, gee, it's twice as likely that it's going to be one copy of the purple gene and one copy of the white gene than it is that it's 2 copies of the purple gene. A little while ago, I told you there are these classic ratios that come up time and again when you're talking about Mendelian genetics. One of those ratios is 3:1. That's the phenotypic ratio that you expect to see in an F2 generation. Okay? There it is, and that's why it happens. Another classic Mendelian ratio is 1 to 2 to 1. And what we're talking about here in this instance is the ratio of the genotypes of homozygote to heterozygote to homozygote. 1 to 2 to 1. Phenotypically, this group and this group are the same, so they add to the 3. Phenotypically, this one and this one, it's an open book. We know what's going on. So let me introduce you, if you haven't heard of this already, to the idea of the product rule. The product rule is an ancient mathematical, statistical kind of principle that lets you say what's the chance that 2 unlikely events would happen simultaneously. And that's what we're trying to wrap our heads around. How can we predict what the chance that 2 unlikely events would happen simultaneously? And the product rule almost always starts to be illustrated with coins getting tossed in the air. And so let's do this, right? I got a silver dollar in my pocket. How about that? Real, genuine silver dollar. Good luck charm of sorts. But, I'm going to toss it. What are the chances that it comes up heads? About 50% if this is a real coin. Hey, I'm lucky. It came up heads. How about that? Pretty cool, huh? So a 50-50 chance of coming up heads or coming up tails. And that's trivial. That's kindergarten stuff. Let's take this to the 3rd grade, okay? What's the chance I'm going to get heads twice in a row? I'm going to do this again. I'm going to toss this coin 2 times now. What are the chances that I get heads both of those times? I got a 50% chance of getting heads the first time. I got a 50% chance of getting heads the second time. 1 and 2 times the product of 1 and 2 times 1 and 2 is 1 and 4. Every 4th time that I toss this coin twice in a row, I should get heads both of those times. Okay? So here, let's give it a try. A loser already. I got tails. All right? Let's see what we get. I got heads. Okay? So, what's the chance that I'll get heads twice in a row? It's 1 and 2 times 1 and 2. And if we put this then into the context of these mating experiments, really what we're talking about is which allele are we going to put into our sperm cells and which allele are we going to put into our egg cells. If you're a heterozygote, every sperm cell has a 50% chance of getting the dominant allele and a 50% chance of getting the recessive allele. It's going to get split right down the middle. Half of your sperm will have the dominant allele, half of your sperm will have the recessive allele. Every egg has a 50% chance of getting either the dominant allele or the recessive allele. They've got to get one or the other, equal mix of both. And here's where the product rule bears out. If a sperm with a capital R, a dominant gene, bumps into an egg with the capital gene, what are you going to get? You're going to get a zygote, a fertilized egg, the beginning of an embryo. You're going to get a zygote. And what's the genotype of that zygote going to be? It's got nothing but capital R's in this particular illustration. We got a fancy word for an organism that has 2 copies of the same allele. We call that a homozygote. It's a zygote, no judgment about what alleles it has, but now we can go further and we can call it a homozygote. It's got 2 alleles that are saying the same thing. Okay? Here, it's a homozygote dominant because both of them are capital R. What happens if a sperm with the recessive allele bumps into an egg with the recessive allele? Once again, we're going to get a homozygote, but this one now is going to be homozygous recessive. And what happens if a sperm with a recessive allele bumps into an egg with a dominant allele, heterozygote, or if you have a sperm with a dominant allele bump into an egg with the recessive allele, heterozygote, voila, right? What we see here is why it is you get that 1 to 2 to 1 genotypic ratio. And a 1, 2, 3 to 1 phenotypic ratio. That's it. That diagram there, not the pennies, but that square, The first guy who sketched this out, after being inspired by Mendel's work and Mendel's talks and writings about particulate inheritance, the first guy who sketched this out, his name was Punnett, and as a tip of the hat to him, we refer to that kind of diagram as a Punnett square. But I think, seriously, that kind of says it all. That's particulate inheritance in a nutshell in terms of how it ends up playing out to give rise to these different phenotypic and genotypic ratios. You can translate that penny diagram from the previous slide directly into what it is that Mendel was working with, and it all flows together and makes sense. What kind of sperm cells, or pollen cells really, are these pea plants able to make? Well, if they're from the F1 generation, the F1 plants are all heterozygous. We know that for sure. What kind of pollen are they going to make? Well, half of the pollen are going to have the dominant purple trait. Half the pollen are going to have the recessive white trait. What kind of eggs are they going to make? Half of them are going to have the dominant purple trait. Half of them will have the recessive white trait. If you bump one of these pollens into one of these eggs, you get a homozygote dominant. If you bump one of these pollens into one of these eggs, you get a homozygote recessive. If you bump this one into this one, you get a heterozygote. You take this one plus this one, you get a heterozygote 1 to 2 to 1, or 3 purple to 1 white. There you go, right? The previous slide was sort of an abstract analogy with coins flying through the air. Now you see genes passing from one generation to another, and it all ties together. There's no other simple way, at least, to describe the results that Mendel was getting with his 3-to-1 ratios. So what can you do with this kind of knowledge? All right? Do we really care how pea plants determine their flower color? Again, this is a tribute to Mendel. He didn't think anybody was going to particularly care. He was just hoping to maybe get a little bit of an insight in terms of how it is that information gets passed from one generation to the next. Are there rules? How do these rules play out? How does the game end up being played in light of these rules? That's all he's after. Didn't really think anybody was going to be interested in how flower color was determined in pea plants. He just wanted to see if there were some rules. And that sort of simple question with a humble organism ends up translating perfectly into the much more complicated and, admittedly, much more interesting, complicated organisms like you and I. And what can you do with this kind of information? Well, if you're a plant breeder, one of the things that you might be able to do is set up an experiment. I don't know that this is a particularly important experiment, but say, and Mendel did say, hey, I got these purple flowering plants in my F2 generation. Which one of them are homozygous, and which ones of them are heterozygous? How am I going to figure this out? And he comes up with this idea. I know what particles, and you and I can say alleles or genes, I know what particles the white flowering plants have. They're homozygous recessive. And I want to know what kind of genotype the purple flowering plants have. I can do an experiment. And I can predict I'll get one of two kinds of results. And depending on which results I get, I'll know what the genotype of the purple flowering plant was. Because a purple flowering plant could either be homozygous dominant or it could be heterozygous, right? If it's homozygous dominant, what kind of sperm is it going to make? It's going to make only uppercase sperm. And if you take those kind of sperm and you hook them up with lowercase, here, we're making eggs out of the uppercase apparently. I'm taking the uppercase, capital P and lowercase p look awfully similar, right? But I'm taking a capital P and a capital P. That's all I can put into my sperm here, and into my eggs here. To the sperm here, all I can put into those sperm cells are the lowercase p. If I hook up a lowercase p with an uppercase p, I get a heterozygote. I get heterozygotes all the way around. the progeny all end up being purple flowering. If all I see as a result of this test cross, of a cross between a purple flowering plant and a white flowering plant, if all I see are purple flowering plants, that purple flowering plant was homozygous dominant, period. In contrast, if it was a heterozygote, half of my progeny will be white flowering and half of them will be purple flowering. And it'll depend upon which of the two versions, which of the two alleles the egg cells got. Half of them will have gotten the purple allele, half of them will have gotten the white allele. All right, so there's a test cross. If you're curious about the genotype of a dominant individual, and you want to go through the trouble, and you're dealing with a dominant and recessive kind of situation, all you need to do is a mating experiment with somebody who's homozygous recessive, and you'll get your answer however long it takes to develop those progeny. So that's the law of segregation. Now, things have progressed a lot since Mendel did his work. And I'll tell you, I think very highly of Mendel's. Phenomenal insights. And I think what really helped him get those insights was rooted in his humility. Really remarkable. But it's not all sunshine and roses when you're talking about Mendel. Everything that he recorded in his diary and everything that he talked about when he'd explain to people about particulate inheritance and then Punnett squares and things like that, everything was always unambiguous, cut and dry, easy to explain. He always talked about monohybrid crosses. He did do some dihybrid crosses, where he would keep track of two traits at the same time, where he would keep track of flower color and seed color. And he'd get things like a 9:3 to 3:1 ratio, the other classic Mendelian ratio. And he would talk about those. I'll show you a slide in a second that shows how that plays out. It's just a slightly more complicated Punnett square. But again, it's not all sunshine and roses with Mendel, because whenever he got a result that didn't adhere to that 1:2:1 or 3:1 or 9:3 to 3:1 ratio, or 1:0 ratio, whenever he got a result that didn't look quite right, you know what he did? He just kind of crumpled up that page and he set it aside and he never told anybody about it. He only talked about the things that he thought that he understood and had a set of rules for. Now, in his defense, that's actually the way that science was done back in the late 1800s. You only presented your case that supported your position. Today, we would have expected him to have also revealed those inconsistent results. And those inconsistent results then give you insights. When the rules don't apply, what else is going on? That's what I'll be talking with you about a week from today. Next Tuesday, we'll talk about the neat stuff that you can get from when those rules are no longer being followed. But let me now take a few minutes to talk with you about some other things that influence heredity. These were things that followed very quickly from Mendel's appreciation of the law of segregation. All right, so let's start. We've already done dominant and recessive, right? Purple versus white flower color, classic example. The first example of a dominant versus a recessive trait. And then that slide from about 6 or 7 slides ago gives you a list of 6 other dominant and recessives. Seed shape, flower position, right? You can fill in the rest of the list if you really care to. So dominant and recessive, pretty straightforward, pretty easy to wrap your head around. Let's talk a little bit about this idea of codominance. And a lot of these names make sense, right? With codominance, what we're talking about is the circumstance where there are 2 alleles, okay? There are 2 versions of the gene, but one of them isn't dominant and the other recessive. Instead, they're equally dominant. They both influence the phenotype of the organisms in which they reside. Here's the classic example of codominance. Did you know that there are these red flowering, true breeding snapdragons out in the world, and that there are true breeding, white flowering snapdragons in the world, those flowers? And did you further know that if you mated a red flowering snapdragon with a white flowering snapdragon, that you would end up with little baby snapdragons, right? What a revelation that is. The surprise might be that the babies aren't red flowering or white flowering. Instead, they're kind of intermediate. You see a little bit of the white trait bleeding through and a little bit of the red trait bleeding through. In a codominant situation, like you have with flower color and snapdragons, both of those alleles end up influencing the phenotype equally. So the F1 generation here is not red or white. Instead, it's pink. And if you have an F1 mating experiment and you generate an F2 generation, look what you get. You get a 1 to 2 to 1 ratio of red to pink to white progeny. How about that? Classic Mendelian genetics kind of ratio, 1 to 2 to 1. Here, the phenotype is something that tells you immediately what the genotype is. Again, the word for this circumstance is called codominance. Straightforward concept, right? With dominant and recessive, the dominant outvotes whatever's going on with the recessive. It wins the day. It's the only thing that you get to see in the phenotype. With codominance, both of the alleles contribute equally to the phenotype, and you blend them together. So that's what's up with codominance. Let's also talk a little bit about multiple alleles. So far, I've only talked with you about characters for which there were 2 alleles: green seeds, yellow seeds, purple flowers, white flowers, wrinkled seeds, smooth seeds, red, white snapdragon flower colors, right? 2 alleles for each of those characters, 2 alleles for each of those genes. There are many genes for which there's more than 2 alleles, okay? Classic example of that is what we see with our human blood types. I'll bet most of you people know what your blood type is. Let me brag a little bit. My blood type is AB negative. Okay, it's the rarest of all blood types. Something like 0.5% of the people in North America are AB negative, but I happen to share it in common with a number of very prominent individuals. Abraham Lincoln, George Washington, right? Are other individuals who are AB negative. What does that mean? Forget the negative part. That's a different gene, the rhesus factor gene. Let's just talk about the AB part of our blood type, right? You know what my blood type means? It means that I've got one allele that says put A antigens on your cells, and I've got another allele that says put B antigens on my cells. What kind of person am I for that particular gene, the antigen gene, that particular gene? I'm a heterozygote. I got an A gene, I got a B gene, 2 different genes for the 2 different versions of the same gene, 2 different alleles. I'm a heterozygote there. But there's a third choice. I have A and B. There is a third allele that's very common within the North American population, and that's the O version. You know what O is? The instructions from the O allele are, don't put any antigens on your cells. So I've got a gene that says put the A antigen on, and I got another allele that says put the B antigen on. There are some people that have an allele that says don't put any antigen on. If you've got 2 copies of the don't put any antigen on allele, we got a name for you. We call you a homozygote, because you got 2 copies of the same thing, but we also call you blood type O, meaning you don't have either the A or the B antigen on your cells. What happens if you've got 2 copies of the A? Well, we got a name for you. You're a homozygote, because you got 2 copies of the same allele. We could also call you blood type A. What if you're blood type B? You got 2 copies of the B allele. But you know what? There's another way to be blood type A. You could be a heterozygote if you had one copy of the A allele and one copy of the O allele. Okay? This is an interesting example of both codominance and polygenic inheritance. There are multiple genes that are involved here. If you have the gene, it shows up in your phenotype, and it influences what your phenotype is, but there's more than just two that goes around. For these blood types, there's 3 different alleles that are commonly observed. So we've done this for most of today's lecture. I've just talked with you about a couple of examples now, snapdragon flower color and ABO blood types, and I've given you one example of multiple alleles, ABO blood types. What's up with this pleiotropy word? Most of the rest of these words, okay, these words I think are English. This, I don't know, is that Greek? It's probably Greek that's got the root there. Pleiotropy. Here's the definition of pleiotropy. Pleiotropy is the word that we use to describe a gene that influences more than one character. Right? So all of the things that Mendel was looking at were single characters genes, right? Flower color, one gene. Seed shape, another gene. There wasn't ever a gene that influenced both seed shape and seed color, or seed shape and flower color. A pleiotropic gene is a gene that influences multiple things, that manifests itself in a number of different characters, a number of different features of the phenotype of the individual. Something of an aside. Well, this actually jogs my memory about something else I should mention to you as well. Did I tell you guys that remember a couple weeks ago they were doing some filming in here and a lot of you guys wore your finest clothes? Tell your parents to watch Michelle Malkin's obscure cable TV show on December 1st. It sounds like that's the date that it's going to be on. So if you were here after that class and you saw the cameras here, that's probably the day it's going to air. That's the last that I've heard. They'll tell me closer to the date exactly when it will occur as well. So anyway, complete tangent of a thought. But I thought of it because I was going to tell you, for whatever reason, a bunch of writers, novelists, and TV show writers pester me with emails. You think you guys ask me a lot of emails? I probably get as many emails from obscure authors as I get from students asking me obscure questions about things. They're kind of fun, though. I enjoy these questions. The writers for a TV show called Rosewood, it's on Thursday nights on Fox, have been asking me like dozens of questions for each episode this season. And so it's fun to sort of watch the show once in a while and say, hey, I'm the one who told them to do that. Or, you know, they needed somebody to die in a coffin-like box without any trace of the poison, and I have to come up with a way to kill people. It's kind of cool. The thing that they've just been asking me about, I suggested that they use a noble gas, right? Because they'd suffocate and nobody would test for the presence of krypton or xenon or something like that. So anyway, there's that. But they've been on a tear lately wanting to have some sort of genetic mystery figured out, and they wanted to have some feature what they really wanted, for some reason, they must have gotten a deal on those contact lenses that change the color of your eyes, because what they wanted to do was focus on tying together some individuals in a family from the fact that they had different colored irises, right? One brown eye, one blue eye, that kind of thing. And so they're hitting me up trying to figure out what sort of gene might be responsible for that. And so I did a little poking around, and I found out that there are 3 or 4 genes that give rise to that particular condition. And one of those genes also is a pleiotropic gene. Okay? And that's why I mentioned it to you here today. There's this Warrenberg syndrome, for which there's a single gene that causes individuals to have different colored irises. But that gene also causes those individuals to, it's a dominant gene, by the way, something of an aside, but it also causes individuals to have white patches of hair, right? There's a fancy word for that, a whitlock? I don't know, but that they have white patches of hair, and they tend to go premature gray, and they also are often deaf in one ear as opposed to the other. A single gene, right, that is affecting multiple characters. Hair color, eye color, hearing capability, not capability, right? So that's what pleiotropy is all about. One gene influencing multiple characters. Epistasis is the word that we use to describe a gene that controls the expression of other genes. So you know what turns genes on and off? We're going to talk about that a lot in the last part of this class, but it's the cliche answer, a protein does it. What turns a gene on or off? A protein does it. Where do proteins come from? Well, they're encoded for by genes, right? And so an epistatic gene is a gene that controls the expression of multiple other genes. Okay? That's another kind of inheritance that we sometimes encounter and need to talk about. Here's an example of polygenic inheritance. Here we've got two genes that are responsible for coat colors in rats, and you can see how you get these, different kinds of rat colors by combining in heterozygotes. Well, when you have heterozygotes mate as your P generation, your F1 generation can have quite a range of alleles. If you have more than two genes, say 3, that's the sort of thing that can give rise to an even greater number of different kinds of progeny. And to some extent, skin's color, pigmentation levels in humans is heavily influenced by 3 genes, and those 3 genes alone can give rise to how many different kinds? 6 different kinds of skin pigmentation. All right? 3 dominant and recessive genes, polygenic inheritance can give you quite a range of features. And so we've got, just as sort of a review, all of these different kinds of inheritance. Pleiotropic, again, is one gene is influencing multiple characters. You can have multiple alleles like we saw with the ABO blood groups, and the rest of the slide speaks for itself. And I tell you what, at this point, I just want to say a couple words to you about this last idea down here, and I'll turn you loose and let you go on, start getting ready, or to continue getting ready for that test. So we've been talking about genetics, right? We've been talking about how information gets passed from one generation to the next. There has been a long-standing debate, and it's not going to be resolved anytime soon, about how much of what we are is determined by our genes as opposed to determined by our environment. I'm sure you guys have heard that in the last year or two, there are people who say they've isolated a gene for dyslexia. And there are people who say they've characterized a gene that's responsible for alcoholism. And they've characterized a gene that's responsible for a predisposition to criminal behavior, right? All sorts of things seem to be influenced by our genes. Twin studies. So when you take identical twins and separate them at birth and have them grow up in different parts of the country and then look to see what's the same and what's different, you expect them to look the same. Did you know you should also expect them to have the same underwear preference? Think about that. All right, boxers versus briefs. There's a genetic component to that. You separate identical twins, you let them grow up in different parts of the country, and then it turns out 90% of the time they have the same preference for underwear. What's up with that? Who would have thought that that choice is something that is governed or influenced significantly by something that we inherited? If that freaks you out, those same twin studies suggest that, and I don't think this is the right word for it, but religiosity, how inclined you are to attend church services, is also genetically influenced quite heavily. You can raise somebody as a Jew and raise somebody as a Catholic for these twins, and it turns out the Jewish guy goes to synagogue regularly, the Catholic guy will go to church regularly. If one doesn't go to synagogue, the other won't go to church. So how much of what we are is governed, is dictated by what we've gotten from our parents, and how much is governed by our environment. Because I'll tell you this, if you got the gene that predisposes you for alcoholism from one or both of your parents, but you grow up in Saudi Arabia where there is no alcohol, your environment is going to have a heavier say in whether or not you're going to be an alcoholic than your genes do. And so it's an interesting give and take and an interesting interplay between our genes and our environment. Something for you to ponder and to think about, interesting things to discuss. That's enough for today. Good luck on Thursday. I'll see you guys on Tuesday.